Welcome to this ARIMA webinar, Decoding Pediatric Cancers, how 3D Genomics reveals promising therapeutic targets in ependymoma. So today we're going to have two presentations. The first will be a short introduction to 3D Genomics from our Director of Research Services, On-Market Support, and Lab Ops. And then that's going to set the stage for our guest speaker, Konstantin Okonyeshnikov, who will share how leading experts from KiTZ and UCSD uncovered novel insights into the molecular properties of aggressive ependymoma brain tumor subgroups using 3D Genomics. And then we're going to wrap up with a Q&A at the end. So please keep those questions coming for a lively discussion after. Now, moving on, our first speaker of the day is Joy Yao, the Director of Research Services, On-Market Support, and Lab Ops here at ARIMA. Joy has a lot of industry experience and worked at a lot of really big companies before coming to ARIMA, and she leads our services team. She's responsible for managing customer projects and overseeing laboratory operations across R&D operations and quality control. And she also spearheads ARIMA's on-market support initiatives that are focused on improving product performance. She received her bachelor's degree from UCSD, and we're very excited to have her here today. Joy, the floor is yours.
Welcome, everyone, and thank you all for joining us today. I am very excited to be here today to share some of the latest from ARIMA Genomics. Today, I will begin with an introduction to ARIMA's technology and how we are able to explore the 3D structure of the genome, before touching on some of the ways that 3D genomics is being applied in cancer research. I'll talk first about how we can use 3D genomics in concert with other omics approaches to explore mechanisms of gene regulation. Second, we'll discuss how to wield 3D genomics data as a tool to identify structural variants such as gene fusions. And finally, I'll talk about how to integrate the identification of structural variants with information about how to perturb gene regulation in the local environment around the structural variant. So let's get this started. To understand the 3D genome, let's start with what we might be more familiar with, what we would call linear genomics. We can define linear genomics as the process of figuring out the order of each of the 3 variants AGCT of the genome in each of our different chromosomes. But of course, there is far more complexity to the genome than can be easily captured in a linear manner. What we know now is that DNA is not just randomly shoved into a nucleus of cells. There is a defined pattern to how chromosomes are packaged into compartments, and this is what 3D genomics allows us to describe. 3D genomics goes beyond providing the genetic sequence information. Its real uniqueness lies in its ability to offer additional insights through the spatial dimensions of the data. Using 3D genomics, we are essentially able to sequence the structure of the chromosome, enabling us to identify structure changes like gene fusions or indels. Moreover, we can use 3D genomics data to measure the interaction between promoters and enhancers, gaining valuable insights into gene regulations. What I want to start with today is the gene regulation aspect and talk about how 3D genomics is an important part of a multi-omics approach to better understand mechanisms underlying gene regulation. In a multi-omics approach, a wide array of tools is available to link the blueprint of the genome sequence to the various epigenetics factors such as chromatin state, non-coding sequence modifications, which in turn exert influence on gene expression, protein abundance, and the interaction between small molecules within the cell. 3D genomics plays a crucial role here in the multi-omics toolkit by identifying long-range genomics interactions that orchestrate gene regulation, thereby helping to bridge the gap between the DNA sequence and the epigenomic factors contributing to the change in gene regulation. ARIMA has developed a number of tools to facilitate investigation of the 3D genome. The core technology underlying these tools is based on HiC, which stands for High Throughput Chromatin Conformation Capture, which was invented in 2009 in Joop Dekker's lab. At ARIMA, we provide kits and services that allow researchers to query 3D genome. The product on the left here is what I want to focus primarily. and that is the genome-wide Hi-C analysis, which, as the name implies, it is to look at the 3D structure of the entire genome. ARIMA also offers different targeted approach to achieve reduced representation of genomic regions of interest, including HiChIP, which uses immunoprecipitation to target interactions of interest, as well as promoter capture Hi-C and custom capture Hi-C, which use sequence-based probe capture to pull down genes and regions of interest to see how the elements organize in 3D chromosome space. The general workflow of ARIMA Hi-C technology begins with sample prep using our kits. ARIMA's kits work with a wide variety of sample types and input amounts, including cell lines, frozen tissues, even FFP tissue that is commonly used for profiling tumors in cancer research. From a given sample, ARIMA's chemistry preserves the spatial organization of chromosomes within the nucleus by cross-linking the interacting pieces of DNA together. That DNA is then digested, tagged, and proximal fragments are religated to preserve the spatial information prior to the NGS library prep. ARIMA's user-friendly workflow are fully compatible with existing NGS library prep kits, easily prepare samples for Illumina short-read sequencing, and provide data output that can be analyzed using ARIMA's other NGS analysis pipelines. So now I want to give you a sense of how we visualize ARIMA Hi-C data. This is a heat map used by the T2T consortium to scaffold the complete human genome sequence. What you are seeing here is each of the chromosomes lined up across the top and along the side. Each point on the heat map represents a physical interaction between two regions of the genome. So the dark red points you are seeing along the diagonal indicates that there are a lot of these 3D interactions between these regions. And the light red, or the white part, indicates minimal 3D interactions. As you can see, it is very common for the chromosomes to form 3D linkage within themselves. But in healthy cells, chromosomes rarely form these linkage with other chromosomes. Let's zoom in to look at these interactions in more detail. What we are looking at here is about 700 kb region on chromosome 2. To help demonstrate how we would interpret this metrics, let's place 4 points along the diagonal. What you probably notice first looking at this is this red dots of the diagonal space. With a high frequency of interaction, what this represents is that the DNA along near the purple point and the DNA at the green point are looping together to give that 3D interaction. To view that in another way, we can represent these 4 points along the linear genome. From the interaction data in the heat map, we expect the purple and the green to physically brought together. And likewise, the gray and the blue are brought together. So we can then infer that the chromosome is organized into domains by these loops that have formed in these regions. So now let's take a look at an example of how Hi-C can be used to gain really important insights into the mechanism of gene regulations or misregulation in cancer. This example was published a few years ago by Klogan and colleagues, and they used Hi-C to look at healthy cells versus cancer cells in a T-cell acute lymphoblastic leukemia patient. Here we are looking at is a zoom-in view of the region near the MYC locus, which is indicated here to the left. And nearby is a known super-enhancer region in healthy T cells. We can see that MYC is contained in an interaction domain that is clearly defined here and is separated from the domain contained by containing the super-enhancer. In TALL, however, the boundary between these two neighboring domains is largely gone, and both elements exist in a single large domain. Looking at some of the associated omics data, we can see that the CTCF binding site near the domain boundaries have been lost. and also an increase in the H3K27 scintillation signal in the area surrounding the super-enhancer. Together, these data suggest that the super-enhancer has been turned on and brought into proximity to MYC, leading to a misregulation of the MYC gene, which we also see gene expression data here. So this is really a nice example of how 3D DNA organization uncovered by KiTZ informs how MYC is regulated in healthy cells, but also give really important clues as to how it is misregulated in leukemia cells. Next, I'd like to shift gears to talk more about how we can use 3D genomics as a tool to identify structure variants, including gene fusion. What really catalyzed our interest and investment into application of 3D genomics data was a collaboration with NYU Langone that began a couple years ago. They had a pediatric patient, a 14-year-old girl with a stage 2 glioma. The tumor was resected and analyzed with DNA and RNA sequencing panels, but no driver mutation was found, so the patient underwent standard chemotherapy and monitoring. About 6 months later, however, this patient experienced very rapid progression, and the tumor returned quite aggressively. And NYU again performed comprehensive DNA and RNA sequencing of the tumor and again failed to identify any driver mutations. At that point, we performed a KiTZ analysis using ARIMA's KiTZ technology, which revealed a novel structure variant near the PD-L1 gene, which encodes a protein that helps immune cells recognize normal healthy cells in the body. In some cancers, high expression of PD-L1 prevents immune cells from recognizing and targeting cancer cells, allowing the tumor cells to proliferate. Since the KiTZ data implicate a possible role of PD-L1 in the patient, our collaborators at NYU decided to look further into that result. And in fact, for this patient, the ARIMA KiTZ result was confirmed with immunohistochemistry staining, which showed a strong diffuse PD-L1 expression in the tumor cell. With PD-L1 expression confirms, the patient was then started on immunotherapy with the drug pembrolizumab. Since the change in treatment, the patient responded very well and her disease is now stable. What is particularly exciting about this case to me is that using 3D genomics, we are able to identify novel structure variant that was missed by other genomics tests and which provide the key function insights that enable the medical team to prescribe the best treatment for our patient. Let's take a step back for a second to understand how we are able to use 3D genomics to identify these structure variant in the genome. If we imagine that we sequence a normal cell, we might get a handful of reads covering a gene located on chromosome A and some covering a gene on chromosome B. And those genes happen to be fused in a cancer cell. Then the linear genomics, you are relying on the sequence reads that span the breakpoints to see evidence of the fusion of these locus. Now in 3D genomics, as we've already seen, we capture information that the 3D structure of the genome. And one really clear characteristic of the structure is that chromosomes most often form these interactions within themselves, but very rarely between different chromosomes. So in the normal cells, you expect to see a lot of interactions in the region surrounding gene A on chromosome A, a lot of in gene B around chromosome B, but not many interactions spanning that distance. However, when those genes are fused together as in a cancer cell, now these genes exist on the same fused chromosome, and we should expect to see many reads spanning that breakpoint. So by using this approach, we are able to leverage the 3D information to essentially amplify the fusion signal to detect these gene fusion. And we are able to do so in a way that is agnostic to the partners involved in the fusion. To show you what the signal looks like in real high-C data, on the left, I'm showing a normal cell line, and we see again that interaction are primary within the same chromosome. By contrast, in cancer cell line, we can see a very distinct off-diagonal signal. So each of these squares is highlighted a 3D linkage or structure variant between chromosomes that is not present in the normal sample. You can see how the amplification of the fusion signal around the breakpoint results in this very clear pattern that we can easily detect to identify gene fusion and the other structure variant. As we've developed the technology, we have done concordance studies to compare ARIMA Hi-C approach to other gold standard methods for identifying gene fusion, such as RNA panels or FISH. And in all cases in which these are known fusions, we are also able to find that same fusion using Hi-C, and then works in a wide variety of solid tumor and liquid tumor types. So we've demonstrated here that we can identify the same gene fusions as existing gold standard methods. But what about other cases like this patient, in which the gold standard test failed to find any driver mutations? Is this case that we started with a unique case, or could 3D genomics be more broadly useful in driver negative cases? Driver negative cancer affects a huge proportion of patients. Almost 500,000 patients in the US alone have tumors with no druggable targets. So we've designed experiments to determine whether we could use 3D genomics to identify druggable targets in case that were classified as driver negative by other gold standard approach. We have analyzed over 200 cases to date that were previously characterized by DNA and RNA sequencing and didn't have any identifiable drug. In 70 of those cases, we have found a fusion event associated with a gene that is a target for FDA-approved therapy already available today. In another 12 samples, we identified fusions associated with gene targeted by drugs in ongoing clinical trials. And in another 29 samples, we identified biomarkers with diagnostic or prognostic utility. In total, we identified biomarkers in 50% of the driver negative samples that could be clinically actionable. Though, as you can see here, the success rate varied depending on the tumor type, most retrospectively, but a few prospectively. We have further tried to validate those results as many samples as we can use IHC, and in a vast majority of these cases we've tested, we are able to confirm their oncoprotein activity, just like we saw in the PD-L1 example. What we're seeing over and over again with these studies is that 3D genomics can really help cancer researchers in 3 key areas. First, 3D genomics can help us detect known fusions that are identified by other gold standard methodologies. But 3D genomics also enable us to find key gene fusions and structure variants that are missed by these other technologies. 3D genomics has really helped to unlock access to fusions from archived FFPE specimens, which can be very challenging in other molecular assays. In the last section today, I'd like to show one example of how we can integrate these structure variant analysis with the gene regulatory analysis using 3D genomics data. Here, I'm showing the interaction landscape around the gene ETV1 on chromosome 7 in a normal prostate cell. And next to it is the interaction between landscape on chromosome 14 in the neighborhood of another gene, MyPol1. In the normal cells, these genes have very little interaction between them since they are on 2 separate chromosomes. Now, if we look at the prostate cancer cells, here we see the interaction on chromosome 7 and the interaction on chromosome 14. In these cells, there is a fusion event between these 2 chromosomes. And we can see the expected interaction between the two chromosomes on the rearranged allele. In the interaction data here, we can see these looping structure forming between ETV1 promoter and other regions in an intron of MYPL1 in a region marked by DNase hypersensitivity. To prove that these regions were functional, the author of the paper removed the enhancer sequences using CRISPR-Cas9 technology and demonstrated a clear reduction of the looping and interaction across the breakpoint, as well as reduced expression of the ETV transcript. So as you can see, this is really exciting case where we can measure directly how the structural rearrangement of the genome can affect the regulation of the genes surrounding the breakpoint. In conclusion, I hope I have helped convince you that there is much more to the genome than just sequence, and that 3D genomics is really important part of our multiomics toolkit. I've shown you today that 3D genomics is concordant with the current gold standard methods for detecting structure variants, particularly gene fusions, and shows a lot of promise for improving diagnosis in a clinical setting by detecting fusions missed by other technologies. Finally, 3D genomics provides unparalleled access to the sequence, structure, and regulatory landscape of cancer genome, revealing novel biomarkers and therapeutic targets. Finally, I'd like to thank many collaborators who have provided us with samples for some of these studies that I've highlighted today. Thank you all for your attention, and I will turn it over to Janetta to introduce our next presenter.
Wonderful. Thank you, Joy, for that excellent presentation. Moving on to our guest speaker, I'd like to introduce Konstantin Okonyechnikov. Konstantin received his PhD in bioinformatics at the Max Planck Institute for Infection Biology in Berlin, where he specialized in developing widely used bioinformatics tools for high throughput sequencing data analysis. Currently, he is a senior postdoctoral researcher at KiTZ within the Pediatric Neuro-Oncology Department. Konstantin's primary research focus is the analysis of massive multiomics data with a specific emphasis on identifying potential medical targets for the treatment of childhood brain tumors. We are very impressed and very excited to have you here, Konstantin, and the floor is yours.
Thanks a lot for this nice introduction. I'm really happy to have an opportunity to present the results of application of high-C technology in investigation of pediatric brain tumors, ependymomas. And in my talk, I will give more details on this, but of course, first, for everyone, the overview of this brain tumor. Unfortunately, it can occur in almost any age and in almost any location of the brain. And initially, it was stated to be histologically as a brain tumor, but during the past decade, the molecular biology allowed to improve strongly the classification of these tumors. So they are separated by the location, either subtentorial, posterior fossa, or the spinal, but also by molecular properties of various factors. And this classification also allowed to identify specific subtypes of these tumors. And actually, in our study, we were focused on two of these subtypes. First one is called posterior fossa ependymoma, and it's quite, unfortunately, dangerous because it has really poor survival. And what is interesting also, this tumor type, it has no information about the genetic mutations. So simply there were various attempts using the whole genome sequence and other data types to find this, and nothing was detected except some significant copy number changes. But what is interesting, this subtype of ependymoma, PFA, has a really epigenetic factor. It's the loss of H3K27 trimethylation across almost whole genome. This is also something similar in other brain tumors. Maybe you heard about this, the gliomas. And also, as the gliomas, this PFA group is the same danger. Another group of the ependymoma that we are also working on is the ZFTA. Actually, this group structure is known for the origin of the fusion with the gene ZFTA. The most well-known fusion is the ZFTA relay, but also it's associated with the chromatripsis in the chromosome 11, where exactly these both genes are located. Again, unfortunately, this tumor has quite bad results in the treatment. And both of these tumor subtypes, both PFA and the ZFTA, they occur in the early age. And according to some doctoral regulations, no chemotherapy, for example, allowed for these tumors due to the side effects. And there is currently the lack of the treatment targets. So the way to find this is exactly application of the molecular biology. And we were trying this using various techniques, especially the epigenetics data in context of the such tumor classes, the PFA. For example, our study from 2019, we focused on usage of K27 acetylation signal to find the enhancers, right? The enhancers that allow to control the genes and to identify the associations between the genes and the enhancers. So potentially, if we find the gene which is associated with several critical enhancers or the super enhancers, it might be important for the development of the tumor and it could be a potential target for the treatment. So basically, in this task, we also did some validations, and once we identified these fusions, and there were some interesting candidates. But what was interesting, how can we understand if the gene is regulated by the enhancer? Is this really some specific associations? And we use a specific strategy for this. So one of the known terms which was derived from the chromosomal temperature and capture data is a topologically associated domain. This is a genomic region which actually remains kind of conserved in the chromatin structure across various cell types, across various tissue fragments, and it shows that the groups of the genes will be lying inside of this topologically associated domains. And to try to find associations between the enhancers of the genes, we were looking for the correlations across the tumors between the genes and the enhancers inside of these topologically associated domains. And yes, some of the results we were validating, and yeah, there are some ongoing further possible investigation of medical targets, but it still remains a little bit unclear. Do we really know that this topological domain is specific for the tumors? And we used in our approach the such a cell line as from the progenitor stem cells, IMR90, but we still cannot be sure that this is a connection really exists. So we were missing this information. And one of the benefits of the high-C data is exactly that we can find direct loops, direct connections between the gene enhancers. And it was one of the main motivations to apply this technique on ependymoma tumors. Another thing that we also were interested in is to look at the structural variants, the topologically associated domains, and combine it with other data types which we also had for the tumors. So in the project, we focused actually on 18 tumor samples from PFA and ZFTA. We looked both on the primary and relapse cases, especially for the PFA tumors, because unfortunately this tumor class also quite frequently occurs with the relapses. And importantly, we had not only high-C data for each of these sample, but a lot of other data types, including starting from a methylation classification, which identifies simply one of the best molecular methods to classify the central nervous system tumors, RNA sequencing data, gene expression, whole genome sequencing data, various types of the chip sequencing data. And what is important also, the complexity to work on these tumors is the also lack of the materials, because right after chirurgy or some other operations in the tumors, there is a lack of the materials that could be frozen. And nevertheless, yes, we managed fully to apply various high-C protocol adjustments based on this. And yeah, looking at this cohort, first thing was simply to perform the analysis. I think you already see this nice figure clearly showing these effects when you have the connections inside of which within each chromosome, while the interchromosomal connections are not so strong. We also had some specific variants across the samples, again, because some of the tissues quality was quite low. And this could be clearly seen when we look simply on the number of the high-C loop connections identified in each of the samples. It might be kind of looking a little bit variable, right? But nevertheless, I will show further that despite this even limitations, we clearly saw everything in a good quality. So yeah, for this data type, we went through the standard analysis. There are luckily already many tools well developed bioinformatics first to analyze the process data, starting from the reads, going into detection of the loops, topologically associated domains, also so-called AB compartments, the segments of the chromatin structure. And already even from this first look, even this variance right across the number of the contacts detected per each sample, we clearly see the specificity when looking at the TATs, how similar are the TATs between these tumors. And as a control, we used also cerebellum astrocytes, publicly available high-C data. And clearly, we already saw that even though TATs, they remain similar across the cell types, there is still variance. And the most similarity was observed across the tumors, across the ependymomas, and the similarity was much lower to these astrocytes. The same will be if you look at the MR90 again. So there's still, even though the TATs remain stable, like 70, 80%, it's critical to look at the particular target, in our case, ependymoma tumors, to identify the correct TATs. And one more clear verification of the full correctness of the obtained data was simply unsupervised clustering of the compartments and the loops, which allowed clearly to not only distinguish the variance between the samples, but also clearly demonstrated the differences between these molecular groups, between the PFA and the ZFTA. Another interesting effect that we clearly observed, and actually we confirmed because it was also previously shown in other studies, that why is it critical to look at the chromosomal conformation capture? We simply looked at the loops identified for each sample and how it affects the expression of the genes. We simply investigated the expression of the genes connected with each other by loops, connected to the enhancers, the ChIP-seq signals that we also had for each of our cases from the HK 27 distillation ChIP-seq data. And simply also we looked at those genes that are not connected either to gene to enhancer. And you clearly can see that based on the expression, this is a strong effect of being in the chromosomal conformation capture in this, the loop connection has an impact in an increase of the expression. It was again another important factor to look at this high-C data. And further, we used the standard approach, again, the algorithm to connect the genes and enhancers to look only on those cases which have the connection between loop. Here is one simple example, right? So we can look at the signals between the gene expression, also with the signal strength of the enhancer, right? The normalized values. And we use the loops. We use simply the loops, the connection to confirm that these genes and enhancers are in interaction. And what is interesting, as I noted previously, we used the MR90 TATs, right, to look into this, while the loops actually verified, yeah, about 75% of our previous result to find such genes that were found. But we also find even almost two times more novel connections which were missing when using these stats. And there are many interesting candidates among them. We tried to also, of course, look at the more details about these genes, but this was already one first step in the application of this high-C data, uncovering novelties in the ependymomas. We further look more into the loops, and we tried to find that there is some specific group-specific activity of the chromatin similar to the genes. And we immediately identified one of the most statistically evident effects. There was a loop, a group of the loops identified only in the PFA tumors in the chromosome 2 and not found in the relays. Okay, we thought maybe this is something connected to the, maybe this is obtained like for the other tissues. And we looked also on available high-C publicly available materials, such as, for example, NPCs or MR90, and we didn't find the presence of this loop. This loop appeared to be even kind of a combination of the contacts in the segment of the chromosome 2 with many genes involved. What is interesting also that we double-checked and we can be confident that there are no structural variants, no changes in the genome which influences. So basically, this is some specific chromatin contact, which is found only in the PFAs. And potentially this could be somehow associated to the cells of origin of this tumor type, simply this normal cell type where the PFA ependymomas are occurring from. Why did we think so? Because on the borders of these loops actually located known biological gene complex, the LX12, HOXD34, which is known to be associated with some parallel processes in the regulation of the chromatin. Moreover, exactly in the end of previous year, when we were already close to finish our study, there was a publication, independent study, which also demonstrates the clear associations of these blocks, of this kind of borders of this loop with the cells of origin of the PFAs. So it was a clear confirmation of this effect. We also looked more in detail inside of this loop. Are there some interesting candidates? And yes, we found actually some genes which were fully fitting as a possible markers and potential candidates for the further investigation that were specific in expression in ependymomas. We also did some experimental validations for example, for 2 of them. And we clearly could confirm using some shRNA techniques and other similar that exactly in the PFA cell lines, this is a sensitive genes. While, for example, our negative control, right, this relay tumors, worth mentioning that typically for the central nervous system tumors, it's hard to get the normal tissue, normal like a brain materials, and there is a strategy to use different tumor subclasses to compare to each other to verify the specificity. And yes, in the relays, we didn't find any confirmation of the for this pair of genes. Moreover, for MAP3K20, we even found the potential inhibitor for this. So again, it could be a possible future interesting candidate for the medical application. As was already noted, right, KiTZ data is also a really nice way to detect structural variants. I'm not going again here right in detail, but yeah, just to know there were already several clear studies that demonstrated that it's beneficial to use, that some of the structural variants that can be detected only with the KiTZ data and further validated, they were not, for example, found with other data type, like the standard whole genome sequence data. And yeah, there are some really nice methods already available for this task, and we also applied it on our cases. And importantly, we had a really good control, right? There's the group, the ZFTA. There is this ZFTA relay fusion, which is caused by structural variants in the chromosome 11. And we definitely, in our cases, we had the verification that these fusions exist due to some structural variants. And we clearly could see the structural variants identified by the KiTZ data. Here's this example on the top, these 3 ZFTA cases, these locations, and clear somatic variants. While, for example, in the PFA, right, you see a clear normal genome. Yeah, we also further looked more in detail on this location. And it was quite interesting because despite variants into the translocations, like in one case, you can have a duplication leading to the fusion, another there is some other type of the structural variant. But what we saw actually a formation of a neotad. So what is this? Simply when we look at the genome reconstructed, when we try to rebuild a novel type of the genome with this, for this particular case, like the 2 duplication sequencing blocks, which are leading to this formation of the fusion, we can also call the TADs, right? And we found in these cases that actually this formation of a novel TAD, exactly in between of this line, this ZFTA relay fusion. But in the same TAD, they also line other genes. And one interesting candidate that we found was ARCOR2. We verified actually in all cases that we had ZFTA that there is a formation of this neotad, and there is also was a statistically evident direct loop between the ZFTA relay fusion and ARCOR2. ARCOR2 is quite, also well studied in other tumor types gene, and also quite specific for the ZFTA ependymomas. And we made into the detailed investigation of ARCOR2. Actually, it's a part of the another epigenetic biological complex with association with other genes, especially SD1 and the HDAC, which has an important part in the regulation of the cell epigenetic structure. And what we started from, we just inspected these genes, the parts of this complex, how important they are for the relay. ependymomas. And yes, our validations clearly confirm this effect, that actually ERCOR2 is specific for these tumors. And deactivating these genes, here's this example of the ERCOR2, here's the SD1, leads to the activation of the cell, while in our control here, the PFA, it has almost no effect. What is interesting, other members of this complex, such as, for example, HDAC, they also might be possible targets for the treatment. Moreover, we did some experiments to look at the activity of the HDAC inhibitor, HDAC1/3 inhibitor, and it demonstrated exactly as a potential possible way to treat these cells, because it clearly was showing the expected deactivation based on the activity. Again, we had the PFA as a control. So simply in identification of this neoTAT inside of ZFTA allowed to find the connection of this fusion to this complex. And yeah, this could be further investigated for the potential medical application. We also looked on the PFA cases to try to find structural variants. Yes, we found there this interesting chromatin to loop, but yeah, we didn't identify any structural variants, even on the high level quality in the primary tumors. Again, it was already known for these tumors based on other data types that it's something like that. Nevertheless, we also investigated the relapses of the PFA tumors. And we basically identified in most of them, again, it is also known, the structural variants, especially these PFA relapses are associated with 1Q gain amplification, which has also really negative association with survival. And this factor was really interesting because we also, even though there were different, we had two cases of the relapses, we tried to rebuild the structural variants, and we found that in both of them, there is the same fragment of the genome attached on this 1Q chromosome part. And we looked basically in this region, what are the genes lying there, and one immediately appeared to be as quite interesting. And this gene is a LAMC1. LAMC1 is specific for the PFA ependymomas. For PFA ependymomas is 1Q gain. It also has some other associations. And based on the reconstruction of the neoTATs, we clearly could confirm that there are some structural effect that leads to appearance of this LAMC1 to formation of the neoTATs with activity of LAMC1. We clearly confirmed this also from the experimental validation, again, with the shRNA, the specificity of the LAMC1 to the PFAs. Moreover, we exactly performed the experiment in the cell line of the PFAs obtained from the relapse, and we could clearly see this effect there. We looked also, interestingly, on some other cases that we had, for example, FP tissues, and we observed that actually the LAMC1 seems to be more activated in the relapses based on expression. We fully confirmed this effect by inspecting additional large cohorts. Importantly, we also tried to look at the HiC data from these FP cases, and despite some quality issues, we clearly identified also the structural variants, such as the complex translocations or duplications where LAMC1 again was lying in the same locations. We tried also further to inspect, like on a global level, this LAMC1 association with the relapses. And for this purpose, we had an extended bulk cohort of large project focusing focusing exactly on the relapse childhood brain tumors. And what we found there, that definitely the LAMC1 was again detected with close connection to the structural variants. Exactly looking even on the three top cases of the relapses with the high LAMC1 expression, we could clearly see that there are some structural variants which support this connection to the blocks touched by LAMC1. So it might be quite interesting perspective target for further investigation. But here, of course, more look into this relapses cases would be required. So this is an interesting direction. And yeah, going further into the epigenetics, as I noted, right, that another quite interesting effect that could be detected from using the HiC data, it's this so-called CTCF loss and the hypermethylation. So basically, what could happen in some cancer activity cells? If there is a, let's say, some established chromatin structure, there is always should be some CTCF blocks which are lying on the borders of the tags, on the borders of the large fragments. It's also well known and studied and proved from many various tissue types and organisms. But what happens if we try to remove this border? If we either deactivate CTCF, remove the activity of CTCF, or make it like hypermethylated, what could happen is that this fragment then opens and we have a novel possible connections formed from the chromatin. And one interesting effect, which was proved in some of the tumors clearly, there are several interesting studies here, I'm showing one example of them, that this opening, the loss of the CTCF and the hypermethylation leads to touch of the enhancer to the gene's activity, leading to the overexpression of a gene. And yeah, this overexpression further really has strong impact on the development of the tumor or the, again, activity of the tumor. And yeah, this could be, again, investigated from the high-C data if we can find something like that in the tumors. Of course, for this, we also need some other data types, such as, for example, ChIP-seq, CTCF, and whole genome bisulfite sequencing. And we had it exactly for ependymoma tumors. And immediately one interesting effect, what we clearly observed, is the specificity of the PFAs. First of all, we clearly saw that actually the enrichment of when we look at the differences between the PFA and ZFTA ependymomas, that much more loss of the CTCF site is observed in the PFAs. We further overlapped the differential peaks with the differentially methylated regions, simply those regions which have different methylation level. And we saw again another increase of the overlap, the specificity of the PFA CTCL of hypermethylation for this comparison. It was clearly statistically much more enriched in comparison to the same effect in the ZFTA. And here just the one visualization showing this, right? So that in the PFA, this is a top differential binding CTCF sites, which are also overlapping with the DMRs. And we can clearly see that there is this strong effect of the hypermethylation and the loss of the CTCA and the PFA in comparison to the ZFTA. So we further use this information to try to find possible candidates. For this purpose, we again use the high-C data, and our interest were possible gene enhancer pairs, which are in the correlation, and there is a loop that connects these genes and enhancer. And what is important inside of this loop and between the loop is lying the segment where the CTCF signal is lost in the PFAs, and also this is a hypermethylation in the PFA again versus the relay. Of course, we tried to use additional information to confirm this. For example, knowledge about this genes specificity in the high global cohorts, in the expression. Maybe there are some other experiments that were validating these genes, such as some possible CRISPR arrays. And yeah, we found actually a list of the candidates. And one of them, here's an example of this AL4C gene. Actually, it was also validated in some of the CRISPR arrays. That's why we actually tried to focus on it more precisely and the demonstrated specificity to the ependymoma tumors. What you clearly can see, there is a gene. It's again specifically expressed in the PFA ependymomas. It has some loops which connected to the groups of the enhancers on the side, which are not present in the relays. And again, there is no loop like this in the relays. And what is interesting, exactly in between, there are these regions which are have no CTCF signal in the PFAs, and they're also hypermethylated. We, of course, wait further into the validation of this observed effect. And yeah, we did similar to other genes, we confirmed them clearly in the PFAs with the shRNA. Moreover, I'm not showing here, but in the relays and negative control, there was no effect. But then the question was, but is it really connected to the CTCF hypermethylation, or it is some other effect? And for this purpose, we actually designed one interesting experiment. So we looked exactly this region, the region of the CTCF loci which was not present, not active in the CTCF peak in the PFAs, but strongly active in the ZFTA cell line. So what we tried to do, can we simply look at the ZFTA cell line, remove the CTCF site, and see what will be effect on the expression of the RFOC? For this purpose, from this region, we applied the CRISPR-Cas. It was an amazing experiment by Alan Campos, and it allowed simply to remove this potential binding site for activation of the CTCF. We further inspected what will be result on the RFOC expression in the cell line, and we clearly could observe the growth of the RFOC, which was previously not active strongly in this ZFTA cell line, that it was very high in expression. So it simply was showing the effect is really caused by this loop. I'm showing only one this main experiment, but we also had some other interesting which further confirm this. And this was only like one candidate, but we also validated similar to another candidate, and the full list still remains as an interesting source of possible future targets. I'll go a little bit into the summary, right? So we applied the high-C data on ependymoma tumors, and we found some really interesting novel things, such as, for example, we again increased the list of the enhancer-associated genes as a potential targets. We found the novel role of the RCOR2 complex in the ZFTA ependymomas, and the direct association with the fusions. In the PFA ependymomas, we found a specific unique loop, which is not present in any other tumors, at least those we are inspected. And moreover, in the PFAs, CTCF hypermethylation effect was found, and there were many confirmations for the interesting candidates based on this as well. All of these discoveries were for this example genes that we selected were validated experimentally, and the future steps will be, of course, to look further into them in the possible medical application. With this, yeah, super thankful to all the participants, especially to Alan Campos, who did all the experimental awesome work, and also to our supervisor, Marcel, and to Lucas. There are many people involved. We are all super thankful to all of them, of course. Maybe I missed some details, then please take a look at the manuscript that is already available online. And thanks a lot for your attention.
This question, I think, is for Kristen, or I'll field it to Kristen. In normal cells, interactions between chromosomes also exist. How do you know the new interaction is a fusion event or just a strong interaction?
Yeah, so I think first I would say that we can detect these sorts of fusion events and other structural variants even within chromosomes. The interactions that we see within chromosomes, and I think maybe you saw some of this in Konstantin's talk as well, have very clearly defined patterns. So we know what a tab looks like, we know what loops look like, we know what chromosomal compartments look like, and all of these levels of organization have these really defined patterns that we see on the heat maps in the high-C data. When we see something that deviates from that, whether that's between two different chromosomes where those interactions are very rare, the signal is very, very low in a normal cell, or in regions within the same chromosome that are outside of those normal levels of interaction, then we can say that those are probably structural variants. And those structural variants too have very clear signals and very clearly defined patterns that are associated with them. So like a balanced translocation, we can see this very clean, pretty butterfly signal that's associated with that. And you can see some of that in maybe some of the heat maps that we've seen today. Copy number variation, deletions, indels, those have their own specific patterns that you can detect as well. So there's some, like, it's not just a strong signal that we're looking for, there is also this pattern of that signal that we can associate with it as well.
This question, I guess maybe Konstantin would want to answer it. I know you briefly showed optical genome mapping as a technique on your slides, but This question is, how does the sensitivity and specificity of Hi-C compare with optical genome mapping and detecting non-canonical SVs?
Yeah, from our side, right? So what we looked at our tumors, this is exactly what we were hoping to find, because it's known that for some cases, only from Hi-C data, it's possible to detect the structure variants, especially in the repeat regions close to the also central MERS that were only possible to identify with data type. Again, so in what we didn't detect anything in our tumor cases, but it's probably because of the nature of this, the ependymoma tumor subtype. But in the, for example, we clearly could detect chromatopsis in this ZFTA ependymomas, which is also was always not possible kind of with, for example, whole genome sequencing, this full reconstruction of the chromatopsis.
So thank you. I know we're about 8 minutes past the hour, and it's getting a little bit late in Europe as well, but maybe Konstantin, since you're here, I would love to just ask a more broad question for you as we wrap things up. What are the next steps? What's your lab focusing on based on this work that you guys recently published? Love to get a little glimpse of whatever you can share.
Yeah, so definitely what we found in some of this tumor subtype, you remember this chromosome on the 2 loop, which is specific for the PFA ependymomas. What we are interested actually to try to prove if it really cells of origin. For this purpose, what we're thinking about to apply the chromosomal conformation capture on the normal cell types. And actually, this is also a big area of research, the brain investigation in the chromosomal connections. There is not much material available on it. And it would be interesting not only for ependymoma brain tumors, but many other brain diseases and cancer types, simply to try to look into this chromosomal structure in the brain, into the cell types, try to look into this in a specific manner. Yeah, ideally, of course, what would be, I think, in the future, the single cell kind of Hi-C technique, which is a very interesting direction. And yeah, this is also what we are thinking about. And in the context of the tumors, if we find these associations, this is also will help further to improve possible the adjustment of the identified targets exactly for the treatment. Can we touch this? Can we use this as a marker of the cell, for example?
Awesome. Yeah, that's very exciting. Really will be great to see what your team, continues to do in this field. So we're at 10 minutes past the hour, and we have a few folks that have stayed with us till the very end. So thank you very much for being here. We're going to wrap things up here. If you have any questions for Konstantin, we will follow up in an email with recording to the webinar, as well as contact information for our wonderful guest speaker. So you'll be able to reach out and ask additional questions if we didn't get to them. And overall, thank you to Kristen and to Joy for your presentations as well. And we're going to wrap it up here. Have a great rest of your Wednesday, everyone. Bye.